Hello. Now we are going to talk about starch. Starch is very unique because it is only polysaccharide that we can find in distinct granules. So what is granule is, if you think about a snowball that's tightly packed inside is filled, and then you can actually form a round shape, and then amylose and amylopectin molecules, they are packed into that very tightly packed snowball. So starch is unique for many reasons, and one of them is because it's found in distinct granules, and the second is it's a combination of two types of polysaccharides or biomolecules. We sometimes use different terminologies, and it's amylose and amylopectin, and we are going to define them shortly. And then starch is embedded into gluten matrix in the dough, so it is very important for the texture of the breadcrumb. We have to be very careful about how much starch is used by yeast during fermentation process, because part of the starch is hydrolyzed during fermentation. And then we need intact starch granules for gelatinization during bread baking. Why? We know the texture of the bread and then the feeling after we consume cereal-based products are coming from bread. And then again, also starch and the changes in the starch chemistry are the main factor for bread staling. So these are starch granules. We talked about them when we were discussing endosperm structure of wheat grain. So we know starch is botanically only located in endosperm, and for wheat starch is unique than corn or rice or other starches you will hear. Why? Because we have relatively bigger starch granules and we call them A granules. And then we also have smaller starch granules and we call them B starch granules. So there are different sizes and different shapes for all the starches based on the botanical source. So if I see this under the microscope, I will say that this is wheat starch because I know it's the distinct look of A and B type of starch granules. But then corn starch, it has its unique look under a microscope, and tapioca and rice. So an expert can actually identify the botanical source based on the shape of these granules. Again, starch is unique for many reasons. Another reason it's unique is because it's semi-crystalline. It's partially crystalline. What does that mean? If you think about your salt you add in your food or sugar that you add in your tea and coffee, those are perfect crystalline, shiny looking molecules. But the starch is in between, it's semi-crystalline. If you think about uncooked rice, it's going to be very tough and firm before cooking. And if we eat raw uncooked rice, we cannot digest it. It's because the starch in the rice is not cooked, it's not gelatinized, it's semi-crystalline. So the enzymes in our digestive tract are not going to be able to digest that enzyme. But with the cooking process, we actually change biochemistry, the chemistry of starch, so it becomes enzyme accessible. And then the enzymes in our digestive tract can digest them and convert them to glucose. Starch is very important for human. Starch is the only polysaccharide that human can digest and convert to energy. And majority of our calorie intake is coming from starch. If you think about the world's diet trends, every culture has a starchy food as the staple food in their diet. For Asian countries, rice. I know some Asian countries, rice is consumed even in the breakfast. Some Asian countries, it's more wheat-based products, bread. I think more and more Western diet, more bread-based products are on the diet. And then if you go Africa, it's cassava and the sweet potato are the major starch sources. So starch is unique because of major calorie source, the way that it is found in the granular form, and it is made of two important polysaccharides, amylose and amylopectin. And for wheat starch, we know that we have bimodal size distribution. And for A-type granules, they will be larger in lenticular shape, and you can see them in the figure with green. They are big. And then the smaller ones are marked with yellow arrows, and these are the B-type starch granules, and they have spherical shape, but the bigger ones, they have lenticular shape. So starch, there is thousands of literature on the what is the starch chemistry and what happens the starch during cooking process. This figure is a nice cartoon, and you can think about the snowball, and if you cut this from half and you look at this picture, so this is like half cut starch granule and what we have inside. So we have the central part, and this is called hilum, and then this amylose and amylopectin, they are added into the granular structure. So here we have growth rings, like we see circles, and then what these circles are, we have the interchanging patterns of crystalline versus amorphous regions. So here the dark color rings are the crystalline regions, and then the light color is for the amorphous regions. So this is why starch is semi-crystalline, because not fully crystalline, it's semi. We have the non-crystalline part and the crystalline part. And then amylose, amylopectin, they are distributed inside the granule. We already talked about starch is made of amylose and amylopectin. So amylose is alpha-1,4 linked glucose. Remember, I said beta-glucan for this beta-1,4 linked glucose, and you have 1,3 linkage for every 3 or 4 glucose. And the cellulose is all 100% beta-1,4 linkage, this passion. For starch, it's glucose molecule, but they are linked 1,4 and is down the plane. An amylose molecule is we consider as linear molecule because it behaves like a linear molecule. There is almost no branches, very low level of branching, and 25% of the native wheat starch is amylose. Amylose is very important for the gelatinization of starch and then also recrystallization of starch. We are going to talk about it. So amylopectin is the bigger molecule. Amylose is smaller in size and linear. Amylopectin, we have 1,4 linkages here, but then there is branching points. Then we have 1,6 branching points. And most of the wheat starch, in native wheat starch, is amylopectin. So 75% is going to be amylopectin. And this is amylose and amylopectin. Again, amylose is linear. And then amylopectin, you see there is lots of branches. And this section, for example, this is still an amylopectin, but this section there is no branching. And then when they are inside the granule, when they come together, they are going to have crystalline behavior for this region. But in the distinct granules, for the branching points, they are going to have the amorphous non-crystalline regions. This is the linear part of the amylopectin, and the branching point is 1,6. And in terms of more into chemistry, for every single sugar molecule, we need to think about if they are going to contribute color formation in the food product. Have you ever thought that why bread crust is brown and the crumb is white? So bread, outside of the bread is brown because of browning reaction. And the browning reactions are results of carbohydrates or sugars. Again, sometimes we use the word sugars in the food formulations. In terms of chemistry, every starch molecule, they have one and one reducing end, and they can have multiple non-reducing ends. If you look at amylopectin, there are multiple non-reducing ends. There is one and one reducing end for each molecule. What does that mean? In the chemistry, if something is reducing, that means it's oxidizing something. But this is the glucose that will be responsible for the color formation reaction. So this is why if you have so many starches hydrolyzed, or if we add non-fat dry milk into baking formulations, you will have darker color. Why? Because of the lactose in the milk is going to contribute the color formation because of this chemistry here. These are important. And then more on amylopectin again. Amylopectin is like a very branched tree. It's very big. And then this is how amylose is actually found in three-dimensional system. So one thing I want to talk about with amylose is amylose has this very DNA-like crystalline structure, and this is important for us to control bread staling reactions. And this is why we add some lipid-like compounds into bread formulations so we can have this amylose lipid complexes. So amylose is very linear. Amylose and both amylopectin, they will be part of the gel formation. And we are going to expand this topic a little later, but amylose molecules, because they are linear, it's easier for them to come together. They are having interactions, and then those interactions are causing firmness in the breadcrumbs. So your bread becomes more tough and drier. It's because of interactions of amylose molecules with each other. Then later amylopectin is part of it too. But if we add some lipids, they actually they penetrate inside the amylose molecules and they form amylose lipid complexes. That's why the pan bread in the United States has shelf life of 10 days. It's going to remain soft because of we are forming this during the baking process. And then again, this is one of the reasons that the lipid complex formation is going to be very important for us to control staling process. And we are delaying starch retrogradation. We are delaying starch molecules to interact with each other. And amylose-amylopectin ratio is very important, and sometimes you hear waxy starch or high amylose starch. So normal starch is typically we have 25 to 30% amylose. If you hear waxy, that means it's 100% amylopectin. And then high amylose is 50 or 70% of amylose content. For ideal bread baking quality, all the wheat varieties that it's over in the market are regular starch ratios, 25-30%. These other ones are going to be strictly controlled, identity preserved wheat varieties. So general properties of starch. Starch is not soluble in cold water. We can have slight swelling, but it's not going to be soluble in water. So you can add a couple spoons of starch into the water and just wait, it's going to precipitate. But think about when you are making pudding, when you add water or milk and then you start heating, we see increase in viscosity, we see changes in that food model, whatever it is. So starch is a great way to control the texture and the viscosity for any food system, including wheat-based products.